Hello, my favorite science students. Welcome to my first video notes. Today we're going to be talking about the scientific method. And on your worksheet that I have provided for you, if you could please copy the notes under the correct category. Okay, so for example, for problem, if you can copy the definition for problem or summarize your own definition of problem based on the notes. Okay, this is review. Most of you should be familiar with the scientific method. We're going to be using the scientific method for all the labs that we do in class, so I just want to make sure you know exactly what the scientific method is. So a scientific method can be done multiple ways. There's not one correct way, but there's always a problem or a question that needs to be answered through an experiment. Okay, and the experiment has to be something that's attainable and something that you can show evidence for. Okay, that's considered to be science. There's different faith and goals and certain feelings, I guess, that people believe in, which is okay. There's nothing wrong with having faith or believing in ghosts or anything like that. The only thing is it's not considered science. Okay, that's why they call it faith. You believe in something without seeing it, and that's not science. You can't. It has to show evidence, and not only do you have to show evidence, that evidence must be repeated in another area or by another person. Okay, so for example, I know a lot of people think the supernatural is to be considered science because you can record ghosts and all that stuff, which is fine. I know you see the shows and they try to record it and have voice recordings and stuff like that, but that cannot be repeated. In order for it to be considered true science, another person or even the same person would have to go back and find recordings again. And as we all know, that doesn't always happen. Not everyone can see ghosts, or not everyone says they see ghosts. So in order for it to be science, it has to be everyone can see it. Just like gravity. You know, if I were to drop a pencil, that everyone can see the pencil falling, no one's going to question it. Whether it's a ghost, not everyone can see ghosts, or it's not 100%, so it's not considered to be science, okay? And so again, the scientific method always starts with either a scientific question or a problem that we need to come up with a solution for. After that, we have to do a hypothesis. Okay, and a lot of people get confused with hypothesis and a theory. Okay, a theory is not a hypothesis. A theory is something that has been tested and has been proven, but the theory can change if more evidence arises. Okay, but a hypothesis is a prediction. Okay, it's I guess an educated prediction of how the independent variable will affect the dependent variable, meaning how the experiment, based on the experiment, what the results are. Okay, independent variable is the experiment, dependent variable is the results, which I'm going to go into the next slide. Please remember that this is a video and I am going kind of fast because I only have 15 minutes. But if you need to pause the video to write these notes down, you can. Okay, just a little reminder. So again, remember, hypothesis is not a theory. Hypothesis is something that needs to be tested to be proven. A theory has already been proven, but it can change if more tests, if more evidence arose. But for the most part, it's already been tested and proven. Independent variable is the actual test of the hypothesis. It's the experiment. Okay, it's whatever the scientist changes on purpose or whatever the scientist manipulates to test the hypothesis. So for example, I show the pictures, you know, chemicals being mixed together and And so maybe the hypothesis is there's going to be a reaction when these two chemicals mix. So that would be your hypothesis. You hypothesize that these two chemicals are going to react. And then the independent variable would be you actually combining the chemicals. Okay, so that would be considered the experiment. So it can be very simple, something like that, or it can be a very complicated experiment that most scientists do. And then the dependent variable would be the results. Okay, whatever the outcome of the test of your hypothesis, what responds to the independent variable. So last slide, the independent variable was mixing the two chemicals to see the reaction. The outcome, like if you look below, for example, this man's outcome of mixing the chemicals was an explosion. So there was not only reaction, there was an explosion. Okay, we're not going to be dealing with any exploding chemicals, but just to kind of get my point across, the dependent variable is the outcome of the independent variable. So the independent variable is the experiment. The dependent variable is the results. And usually, as you can see my little chart paper, my bar graph below, usually it's something you can measure. Usually it's something that is measurable, and you could even graph it or put it in a table and data. That's usually the dependent variable, okay? But not all the time. But it's always whatever the outcome of the test is, that's a dependent variable. Most experiments, still talking about experiment here, most experiments have a control. Not all experiments have a control, like for example the mixing of the chemicals, there's not really a control there. But what a control is, it's used in experiment for comparison. So if you can remember Control C for comparison, that's what a control is. So whatever we're doing to the experiment, to whatever we're doing to test our hypothesis, whatever we're doing for the independent variable, that variable is not applied to the control. Okay, and it's not applied to the control on purpose because we're using that control to compare the experiment. So a long time ago, I did an experiment in college, and I took botany, and I had to do any tests I wanted, but it had to be on plants. Okay. So I decided to add vitamins to one plant and see how it grew, and then I added caffeine to another plant and see if it affected its growth. And then I needed my control plant, and in my control plant, I didn't add any vitamins or caffeine or any of my experimental variables to it. Okay, and I used that so I could compare. Okay, because how do I know the vitamins, for example, help the growth of the plant if I didn't compare it to my control? Or how do I know my caffeine hindered the growth of the plant if I didn't compare it to my control? Okay, so that's what a control uses. It uses evidence to show that the experiment did or did not work, as you can see when you compare it to the control. A constant is something that has to be kept the same. I don't know if you can see it, but it's something that has to be kept the same in all trials of the experiment. Okay, so constant is different than control. It's just constant that's something that we need to keep the same. So like for example, in my plant experiment, I needed to keep all the soil the same, the pot the same. I needed to keep the area the same. I needed to keep the plant the same. I needed to water them all the same. And I do that because how would I know the caffeine hindered the growth of my plant? What if I just watered it less than I watered the vitamins? Or what if I watered it less than the control? How would I really know the caffeine hindered it if I didn't water them all the same? I gave them the same water, gave them the same sunlight. So those are called constants, something that's kept the same in the experiment on purpose, so you can test your hypothesis. So in this case, I tested the vitamins and the caffeine. So I needed to keep everything else the same, including the plant, because how can you compare a tomato plant to a flowering plant? Okay, I couldn't be able to do that. The plant would need to be the same. So that's called a constant. Okay, and then I don't have a space for you in the worksheet that I gave you, but you can just write it under control or wherever you have space what a constant is. It's pretty important for experiments. And then last but not least, your results. And your results, again, that's the dependent variable. It's the outcome of your experiment. And there's two types of results that you can have. You can have a qualitative data, which meaning you're kind of observing. It's not something really you can measure, but it's an outcome that you can obviously see and observe, like whether there was a reaction between the chemicals or not. Or it can be quantitative data. And quantitative data is something you could measure, like with a ruler, with a timer, a stopwatch, a weight, a mass, and it's usually something you can measure and you can graph. So, for example, for my experiment, I measured the height of my flower. I measured how many flowers were blooming and how many petals were growing, and I could measure the mass of my flower. I mean, that would be considered a quantitative data. And then I could record my results and make bar graphs, make any graph that would help you. We use graphs to show our data, and it's easier for us to read, and I'll go into more detail on that when we do our labs and our experiments. So just keep in mind, we call qualitative data we observe. Quantitative data is usually data we measure. And results, again, are used to analyze your data from the experiment, take a look at your data, and then you kind of show your results, or you kind of explain your results more in detail in your conclusions. So in your conclusions, usually you want to tell whether your hypothesis was correct or incorrect. So I could say, I originally hypothesized that the vitamins would help the plant grow. And in that case, it actually did help the plant grow for some reason. I don't know why. And then you kind of reflect on why you think that is, and then any other testing that needs to be done to ensure that your hypothesis was correct. Any changes you would make, maybe I would use a different type of soil or something like that. And this is the place that you would put it. And then you would kind of talk about any further testing that we need to be done based on the results that you got. And then if there was any errors in your testing, okay, because in science, errors can occur, and you have to report it. Okay, you can't hide the errors that occur, because that would make your testing invalid. Okay, so you can have errors in science. There's errors in science all the time, but if you have an error in the conclusions, that's where you would say. So I had an error in my plant experiment. I accidentally planted tomato plants instead of flowering plants, and we were supposed to test flowering plants. So I had to take the tomato plant out of the soil and then replant. So it could have affected my data. I feel like it kind of did, because tomato plants take a lot of nutrients from the soil. And I think they affected my soil because the nutrients in my plant, I don't think the soil was real soil, because I had already planted the tomato plants, and I think it took nutrients, and then it affected my ultimate plant growth, because a lot of my flowers did end up dying. So that's where I would put it. I wouldn't hide it. It's just part of science. Sometimes errors occur. Okay, so you're welcome to rewatch this video if you need to, and if you have any questions or concerns, you can ask me. Okay, very quickly, we're going to do a new assignment called the Mythbusters assignment. So if you can please copy this URL in your notes, or copy it into your URL in your iPad or your laptop, you should see something that looks like this. So you need to watch one of these clips and then label the problem from the clip, the hypothesis from the clip, the independent, dependent variable, and results and conclusion in your worksheet on the back.